All right, hi everybody, welcome back. So here we are. This is going to be the second video. So if you've already watched, you should have already watched part one. That was all about transcription and translation, about that relationship between DNA, RNA, and protein. This video is going to talk specifically about RNA. So if you're curious about what RNA is and what all those different kinds of RNA that we just talked about in video one are, this is where you're going to find out about those. So stick around. So once again, this is about that second learning objective. You can compare and contrast these different types of RNA and what they do, basically speaking. So what is RNA? So RNA is another type of nucleic acid. So it's very similar to DNA. So remember, DNA stands for deoxyribonucleic acid. RNA stands for ribonucleic acid. So DNA would have deoxy in front of ribonucleic. But they are very, very similar. They're both long chains of nucleotides. So remember, we talked about nucleotides a couple weeks ago. Nucleotides are made up of a phosphate group, a sugar, and a nitrogen base. So the thing that's different between DNA and RNA is that sugar. So depending on the type of sugar they have, you're going to have either DNA, if that sugar is deoxyribose, so it's missing an oxygen here, or if it's ribose, if it has that extra oxygen. So this is the sugar that we find in RNA, and this is the sugar that we find in DNA. Another difference between RNA and DNA is that RNA is single-stranded. So it's just one single strand of nucleotides, whereas DNA has two strands in that double helix formation. RNA also uses a different nucleotide for some of its letters than DNA does. So remember, DNA uses adenine, thymine, guanine, and cytosine. RNA will use the nucleotide uracil in the place of thymine. So you'll never see any T's in an RNA molecule, but you will see a lot of U's. And RNA is different than DNA because it comes in lots of different shapes and does lots of different functions. DNA is always going to be in that double helix, and it's always going to be that place where we find our genetic code, our genotypes. So a little bit about each of those different types of RNA will come next. So here we have mRNA. So mRNA stands for messenger RNA. It's the type of RNA that's going to carry that genetic material, the genetic code, from the nucleus into the cytoplasm. So remember, in transcription, we take our DNA and we rewrite it as mRNA. This mRNA can fit through these little pores in our nuclear membrane. And so the mRNA can actually get out of the nucleus and into the cytoplasm. DNA is too big. It can't fit through those pores, so it gets stuck there. That's why we need mRNA as our messenger RNA to get that genetic code from the nucleus to the cytoplasm. One thing about mRNA is we're going to talk about codons a lot when we talk about RNA. So every 3 nucleotides, every 3 bases in RNA are something called a codon. So 1, 2, 3, this particular section of the mRNA right here could be one codon. The next 3 could be another codon, and so on and so on. So 3 bases equal codon. This is kind of like when we were talking about our DNA as a recipe, our genetic material as a recipe for who we are. The words of that recipe are going to be 3 bases long. We call those words codons. There's another kind of RNA called rRNA, or ribosomal RNA. This is the RNA that kind of bonds with some proteins to make a structure called a ribosome. So here is a ribosome, and you can see on the inside there is some RNA there. This is rRNA, or ribosomal RNA. So its basic function is that it does form those ribosomes in the cytoplasm. So here's a picture of an RNA or a ribosome, and this is just a fancier picture of a ribosome showing the different proteins in pink and green and the RNA in blue. So you see how they interact, they mix together, they really bond together to make that ribosome structure, that thing that's going to read our mRNA to figure out what amino acids we need to make our proteins. Another type of RNA that we've talked about, so far we've talked about mRNA and rRNA. Now we have tRNA. So tRNA stands for transfer RNA. Its major function is to bring amino acids into the ribosome based on what the ribosome says it needs. So each tRNA has an anticodon that is complementary to a codon on an mRNA molecule. So as the ribosome reads that mRNA, it reads specific mRNA codons, and then it makes signals that tell the cell to bring tRNA in that matches that codon. So every mRNA codon, so every codon, every set of 3 letters, has a tRNA anticodon that goes with it. So attached to this tRNA, so this whole thing is tRNA. Here's the codon, the anticodon. This part's tRNA. Up here, we have our amino acid. So this tRNA transfers those amino acids from everywhere else in the cell onto the ribosome so that it can join that chain of amino acids that make up our proteins. All right, this was a really, really quick overview of the RNA molecules and the different kinds of RNA that we see in transcription and translation. You guys are doing awesome. Keep it up. Have a great week. And I'll see you later.